Dr. Carl Rothfels and from Berkeley. The reason why I say right was because he got his PhD at Duke. And after he got his PhD, and Carl did his postdoc and back in his home country, University of British Columbia. And Carl works on a group called Inferring. And I don't know if you know what that is. And Michigan used to be a major institution and put out a lot of graduates studying that group. So if you don't know, and there's a picture there, and then during the seminar, I have tell us more about it. Although his seminar is about inferring evolution timescale of vascular plant evolution, so I will hand the podium over to Carl, so he will tell you more about it. Okay, welcome.
Thank you very much. Thank you to YQ for inviting me and for organizing what's been a spectacular visit so far. It's a real pleasure for me to be here for a number of reasons. This is a hotbed of fern research, or was, and I understand that Florence Wagner is still with us, and still, I don't know if she's coming in, but 100 years plus of teratology is a hugely impressive accomplishment. And there's other sneaky expats hiding around, like Tony Resnicek, who was, I don't want to embarrass him too much, but was one of the guiding lights that brought me into botany in the very beginning. And in fact, the last time I was here was a little bit over 15 years ago when I interviewed to join Tony's lab to work with Tony for my PhD. So we'll just leave that story at that. It's a real pleasure for me to talk about a project that is a very new one for me, completely hot off the presses, and you'll see exactly how hot off the presses it is as I get a little bit more towards the results section. I should preface this by saying that, as YQ alluded to, I'm not a native Michigander. I'm from across the water in the Northland of Canada. And if I lapse into really strong Canadian, or if I talk just simply too quickly, please feel free to ask me and to slow down and ask questions. I would welcome being interrupted, especially if I'm not being clear. The project today I'm going to talk about is about inferring the time scale of vascular plant evolution. And this is a project that I was very lucky to get some funding for with 3 co-conspirators, so Cindy Loy and Natalie Nagalingam and Michael Sundu. And we are delighted to be able to pursue this project. And of course, as PIs, we're not the ones who ultimately are doing much of the work. And so that particular credit belongs to 2 fantastic postdocs, Dori Contreras and Mike May. And so a lot of the work that I'm presenting today has been due to the hard work of these 2 people. I have that little sort of hidden, obnoxious, old English subtitle. And ultimately, this talk is going to be more about the subtitle than the bigger title, and specifically the lessons learned part. So this is going to be a combination empirical talk where I talk about some actual results, but it's going to be more about some of the methodology. So it's a strongly methodological talk, and I'll try to take it, I'll try to be as engaging about that as possible, because that's the part that I'm really hoping I will get some feedback and some thoughts from the people here today. So vascular plants, they don't really need an introduction, but I should say so anyway. There's a hugely important group. It's very species rich. It's also very disparate, encompassing a huge spectrum of morphologies and ecologies. They are fundamental for the origin of terrestrial life. So basically all life on the surface of the Earth has an intimate connection with plants. Very important for the project I'm going to outline. They have an extensive fossil record. And of course, they're of unparalleled economic importance. Everything we eat basically was a plant or still is a plant. And despite this, you would think that we might have a strong grasp, despite all of these attributes, a strong grasp of their evolutionary history. But we don't. The time scale for plant evolution is hugely contentious. And this isn't specific to plants, but I'm going to highlight plants in particular. Don't pay too much attention to the details here, but across the x-axis are different taxonomic groups. And across the y are the inferred ages from those groups in different studies, highlighted with colored circles. And you can simply see that some groups have hugely uncertain age estimates, spanning often 50 to over 100 million years of uncertainty in the estimates for the age of that group. And why is this? Well, one reason I'm going to argue is based upon our classic way of attaching timescales to phylogenies. And this is going to sound a little bit like a a false start a little bit later in the talk, but I think it's an important element, and I'll come back to it a little bit later. So I want to sort of hammer this home very quickly. As phylogeneticists, we often deal with structures like this. We infer a phylogeny, and we say, OK, this beautiful thing with all these nice abaxial sporangias over here, these weirdos with little flaps around their reproductive structures over here, half a cone, et cetera, and you want to attach a timescale. There's no information in this phylogeny to attach time, because we can be pretty good about estimating the lengths of these branches, but they're in the number of expected, in this case, if it's a molecular phylogeny, expected substitutions per time, per site. And so rate and time are completely confounded. You can get this many changes with a really fast rate and a really slow time, or a really slow rate and a really deep time, and there's no way from these data to tell those two things apart. You need some sort of exterior information to be able to attach a timescale to this phylogeny. And the go-to source of that information is from the fossil record. So you can imagine that you have a fossil that looks like this, that was this old, and we're going to use it to calibrate this phylogeny. We could say, well, it has those 5 little flaps, and so we're pretty sure it's closely related to these two things. But those 5 flaps, that morphology could have evolved here, but it actually could have evolved anywhere along this branch, quite reasonably. And so the earliest that we might think that that morphology evolved is at this node here, which actually is this node right here. And so, whoops, sorry. I'm going backwards or forwards? Yeah, so at that node right there. And if that morphology could have only have evolved at the earliest at that node there, but we know that it exists at this point in time because we have a fossil of it, then we can say, well, maybe we could calibrate our phylogeny like this. There's no way that that node can be any younger than that fossil. It's the earliest that morphology could have evolved. That morphology already existed. There's no way that this node can be younger than that fossil. So that's a minimum age of our tree. But this is a little bit weird, right? We probably don't think that this fossil, this morphology evolved exactly here, and then was immediately fossilized, and we recovered it. We probably think that this is a minimum rather than the actual estimate of the age. And so we might think that, well, I'm more comfortable with a distribution of how old that node is. And so we might make a distribution like this. And this particular distribution implies that we feel relatively confident that the node, like there's a good probability that that node is this old, the peak of this curve, which sort of implies that this fossil was somewhere around here in the phylogeny. But it also leaves open the possibility that node could be all the way down here, implying that the fossil is at this point in the phylogeny. And obviously, those 3 calibrations result in very different inferences of time. So you can imagine that the inferences would be uncertain. And more important, I would say there's a series of other problems that this method suffers from. And so first of all, each fossil has to belong to a particular node. You can't just put a fossil anywhere. And so there's relatively few places, for example, where we can attach the information associated with this fossil. It has to be either here or here or here. You can't put it, for example, somewhere in the middle. So there's not that much information in the fossil that we can use. It has to attach to a node. Only the oldest fossil that is assigned to a particular node can be of any use because they offer minimum information. So if we had a bunch of fossils from this area, we wouldn't be able to use them. They wouldn't provide any information. The assignment of the fossil to the node is based upon the researcher's opinions, and that could be a very well-founded opinion, but nonetheless, you have to make a decision about which node a fossil belongs to. And so we said, well, clearly this has 5 petals, so it belongs up here with this guy's, but we could be wrong. And if we're wrong, our subsequent inferences will be wrong, and we will have no way of knowing. We have to make that decision, and all of our analyses downstream are dependent on that decision. And finally, the relationship of the fossil to the node, so we know which node it attaches to, but the actual relationship temporally with that node is something else that we have to make statements about. We have to decide exactly what sort of distribution to use. And we pick this random one here. But there's many other options that might be available, and it depends upon what we think the fossilization process is like, as well as what we think about the morphological evolution. And that's, again, a statement we have to make. We have to make that constraining distribution. We can't infer it from our data itself, more or less. There is, however, a way that I'm going to argue solves a lot of these problems. And it's this idea called total evidence dating, or TIP dating. And in this approach, you take a matrix of morphological characters for the fossils, but also for your extant taxa. And you combine that together with a matrix of molecular data from only the extant taxa. And you can analyze those together, which will allow you to co-estimate the phylogenetic position of the fossils, the model, the patterns of morphological evolution, and the timescale of the phylogeny simultaneously. And you will get something like this. So instead of treating this as a calibration that we attach to a node, we treat it as its own observation, its own source of data. And we infer all of these patterns, all these components, the phylogeny, the patterns of morphological evolution, and the time scale simultaneously. And so we're excited about this approach. And I should also say that it allows us to include some biologically reasonable, some models with biological meaning for how the tree shape evolves. And these are called fossilized birth-death process models. And the idea being here is that the trees that we observe feature a combination of speciation events, such as the one that gives rise to these two lineages. They include extinction events, such as the ones where these lineages went extinct. But they also include fossilization events, where fossils are sampled. And so if you have a process that models the rates of those three types of events, you can get a reconstruction like this, which is equivalent to what we are ultimately after, which is a tree in which you have fossils that are terminals on tips. You have extant taxa as well, and you have the divergence times for all of your phylogeny. And it's important to highlight a somewhat interesting corollary is that you can actually have sampled ancestors. So in your sample, in your fossils, for example, you can have taxa, you can have accessions that are directly ancestral to other samples in your taxa, in your taxon set. And so we decided that we wanted to use this approach to build a timeline for vascular plants, which is a colossal endeavor. It's a hugely diverse group, as I mentioned, has extremely deep timescales. And this almost certainly is a somewhat foolhardy thing to have proposed, and maybe even more foolhardy for them to have given us money to do. But at least we had the sense to start off with a smaller section, do a pilot study, just to sort of test out the waters and see how this approach is going to work. And the group that we decided to do that in is called the Moradiales. It's an order of eusporangiate ferns. There's about 100 of them currently in 6 genera. They're often called the king ferns. Some of them are huge. They have probably the heaviest leaves in ferns. They have a number of really unusual features. They're sporangia. They have eusporangia, so they have very thick walls with very many spores, quite different than most ferns. And they are fused together in these little sort of packets called synangia. So that's a characteristic that unites the entire order. They have these weird pulvini, these weird sort of flex points on their leaves and their leaflets, something you see very rarely in ferns. They have a unique vasculature. So this is not I realize it's not necessarily clear what is so special about this, but this is a pattern of vasculature you don't find in any other ferns. And they're really quite spectacular and truly bizarre ferns. So this particular genus, for example, there's the synangia, all those fused sporangia, and they open by apical pores, which is not a feature you otherwise see in ferns. They often have quite complex leaf architecture. So here's a more divided species, really quite beautiful. And they have some real weirdos like this thing, which you might not think is a fern at all. It's got reticulate venation. Doesn't look very fernish until you flip it over, and sure enough, oops, whoa. It has the same synangia, so those fused sporangia, except they're radially symmetrical now. It also has what are putatively the world's largest, or among the world's largest stomata. So each of these things that you can see with your naked eye is a stomate on the underside of this plant. So this is another member of the Moradiales. But more important for our purposes, they were formerly ecologically dominant. They're one of the main components of the Carboniferous coal swamps, and they have an extensive fossil record. And there happens to be a data set that was produced that included both morphological and molecular data that we could base our study on. So this seemed like an obvious candidate for an initial study. And so what did we do? Well, we assembled a data set of about exactly 83 accessions. Slightly over half of them are fossils, and the rest are extant. We have a molecular data set for them of about 5,000 sites, and we have almost 100 morphological characters. So this is our core data. And what I'm going to show now is the sort of a schematic of the model of the analysis that we did with these data. And the first thing is that we need to have a model of the tree. So what kind of process gave rise to the tree? And we did three, although we're too hot off the presses to have number 3 yet. So I'm going to only talk about number 1 and 2. So 1 is a uniform prior on all possible trees, and 2 is one that's produced by that fossilized birth-death process that I mentioned a few minutes ago. So effectively 2 tree models. We have 3 models of morphological evolution. Obviously, the evolution of morphology is a complicated thing. It's very difficult, and so we were concerned this might be having a big effect on our inferences. And I'm happy to explain what any of these are to anyone who's interested. Effectively, this is the simplest one that we can have, often called the MKV model. It predicates that the shifts between different states happen in a symmetrical way, and the frequencies of all states are the same. Up to more complicated models, this one is a mixture model of 5 what are effectively F81 mixtures, and I can explain that to anyone who is interested. I think these are actually really important and kind of exciting because morphology is what's underlying all of these inferences. That's the only information that's available from the fossils aside from their date, and so we really are paying a lot of attention to how to model morphology. We have a clock model or a rate model, a model for how the rates of change will change over the tree, the branches of the tree. And we have a separate one of those actually for each of the morphological and molecular data sets. And finally, we have a molecular substitution model, which is a classic model that we apply in molecular phylogenetics all the time. So these are the components of the analyses of the model that we're going to run. And we're particularly interested in these 2 parts. So the morphology model, as I mentioned a minute ago, and the tree model, because we're concerned that these 2 parts may be having a strong influence on our ability to infer this time scale. That's all. Okay. So what are our results? Well, the uniform tree prior looks relatively good. Here we have the 6 genera of the extant Miratiales, 1, 2, 3, 4, 5, and 6. They are related to each other in ways that we think. They are also related to our outgroup in ways that make sense, and the relationships in our outgroup make sense. There are a few problems. This is a lot older than we probably think is reasonable. So we're inferring a crown age of the Miratiales of about 200 plus million years ago. We probably think that's more like 150. And the crown ages in our outgroup are also off, but I'm not as worried about that. And also there's an interesting problem of rooting. It's commonly understood that it's hard to root trees using just time information like this. We didn't attach an outgroup here. I mean, I'm calling this the outgroup, but it wasn't designated as such. The rooting comes from the model itself. And so they got it wrong. Basically, a bunch of these things, these fossils here, the Scolopters, this should be over here with these other ones. So the real root should be somewhere like here. But all in all, quite encouraging. Very pleased, given the very preliminary nature of these analyses, to have a relationship that makes sense like this. I should also say that all of the things from this branch on up are the Miratiaceae, the family that has the extant Miratiales in it, the only family to have extant Miratiales. And these things over here are often put in a different family, the Ceroniaceae. So even our broad patterns are kind of making sense. So we're feeling good today. We see this tree, we're like, all right, this is working out fine. Let's see what the tree is. And I should say that this is from the uniform tree prior. Let's see about the tree that we're most excited about, the one from that fossilized birth-death tree prior. And we look at that, and it looks completely different in ways that are effectively implausible. So it has an age of the extant Miratiales of like 20 million years ago, this needs to be at least 150 million years. So we're off dramatically, catastrophically. Here's all of the extant leptosporangiate ferns. So this is something like, I'm going to forget the exact number, probably over 95% of extant ferns. It's given a crown age of less than 50 million years ago. This is simply impossible. All of our Moradiales fossils are falling way out here, not related to the extant Moradiales. Everything about this is wrong. This is not everything, but this is a deeply, deeply flawed analysis. And so this has been my favorite emoji, my emoji favorite of late. What is going on? And one of the things that I'm excited about is we've also working on some techniques for understanding what is going on. These models are all relatively new, and their behavior isn't all that well understood. And so one thing that we can do is we can use a tool called posterior predictive simulation. And basically what that involves is we take a sample from our model, and in this case we're sampling from the simplest of our morphological models under the uniform tree prior. We take a sample from that model and we simulate new data with the posterior parameter estimates from that model. And we take a look and see if the new data we simulated, do they look like the data that actually generated that posterior. In this case, we're using parsimony score, which is in effect a measure of tree length. And what we see is that the data that we simulate from our posterior estimates are not at all like the data that the data that are our original data. We would expect this curve to include the zero length, zero value, and they don't. So this model is highly inadequate. It generates data that are unlike the data that were used to estimate those parameter values. And if we take a look at the slightly more complicated one, slightly more complicated morphological model, I don't really know whether that's an improvement or not. We get a lot more variance, but perhaps the mean is still a little bit worse. When we look at our final, more complicated model, and it's looking a lot better, but it's still inadequate. It's still not able to produce the empirical data from which those posteriors were estimated. So our morphology models are not capturing what's actually happening in the evolution of those data. And so that might be what's causing our problem. Maybe our morphology models are sufficiently bad that even our best one isn't able to capture what's going on. However, if we overlay now the results from the other tree model, so the fossilized birth-death tree model are in the light shades and the uniform ones are in the dark shades, we see that they are nearly identical. So both models are producing not only equally bad data, but pretty much exactly equally bad data, exactly the same sorts of data. And this is weird. Why would two models produce effectively exactly the same type of data under simulation? Especially when they have huge effects on the trees. So this is a summary of what the trees look like. This is a lineage-through-time plot. And so if we follow the uniform tree prior, this has been simulated. Oh, this is based on 100 samples from the posterior. You see that around 500 million years ago, there's always only one lineage. But come around 400 million years, there's usually two, et cetera. These shaded squares are the 95% credibility intervals. So there's a lot of lineages built up gradually, whereas under the other tree model, there's no overlap. At no point in time, inferred time, do these models produce estimates of the number of lineages that would be present that have their 95% confidence intervals overlapping. So these are extremely different estimates from these two different models. And so how can we have two different models that produce pretty much the same data, but when you simulate under them, but give it extraordinarily different inferences when you use those models for inference? Well, it's because of something we already know, what I already mentioned, which is that relaxed clock models have an identifiability problem. You have rate and time always confounded, and so rate and time in various combinations can produce the exact same data, and the only way to pull them apart is by having some model component that does it. So our model is what's allowing us to differentiate between fast branches and short times versus long branches and long branches and slow times. And we happen to have a very weak, relatively uninformative rate model, so there's not much information there. And so that means that ultimately, the decisions about how the model allocates those two different components is coming from our tree model. And so the uniform tree model happens to prefer really, really deep divergences, and the fossilized birth death, for reasons I can't describe to people who are interested, is pushing us towards really, really recent divergences. So a classic problem of what's called model sensitivity, a prior sensitivity, or model identifiability. So where does this leave us? Well, I want to take a step back for a minute to say that although that may sound a little bit bleak, I'm still hold by the statements I made earlier, which is that this approach, all the problems I've just mentioned, those also would apply to a node dating-based approach. But all of the advantages that I outlined at the beginning still hold. We still have a way to avoid all the challenges that I mentioned about node dating. So that's good. And we have the advantage of being able to use these models that have a little bit more biological realism. And that gives us some power to, now that we know of our problem, we know that this whole class of problems is prior sensitive. It gives us an opportunity to select and to tune our priors to forms that are biologically reasonable, that actually meet our biological expectations. And so I think that it gives us a whole new way of approaching these problems that with node-based dating, simply you don't have it. All of that happens based on the shapes of those distributions, and you have no biological way of understanding what's going on or what's implied by your model. And the other thing I'd like to say, sort of on the positive note, is that I'm really excited about the fact that we also have now a set of tools that allow us to to look under the hood and see what are the problems, what are the things that are driving our inferences. It's very tempting to take a data set and take a model and apply that model to that data set to get an answer, and that answer will have uncertainties, for example, but just think that is done, that you have your answer. But what I've just shown you is you could do that with two different approaches, each of which will give you a full measure of the uncertainty, but they're contingent upon those models being correct. And it's very important to have methods available to diagnose problems that might be happening in your modeling choices. And I should also mention, just as a sort of a little side technical detail, that another way a lot of this is often done in a Bayesian context is by the computation of Bayes factors, which is a way of comparing model fit. Bayes factors are extremely sensitive to the prior in a ways that doesn't really have a biological meaning. So this is in a way of actually looking at the adequacy of the model. How well does that model explain your data? Not simply does that model fit your data better than some competing model. And so I'm optimistic. Maybe I'm optimistic by nature, but I'm kind of excited by the ways that these models are failing us. I think there are ways that we can address. And I think there are ways that have a lot of relevance far beyond, obviously, plant phylogenetics. There's a great need and a great interest in the ability to attach timescales to evolutionary events. We have some powerful tools to do that. And I think that we're demonstrating that our tools are improving, but we still have a long way to go. And there's still some real pitfalls that as an evolutionary biology community, we need to be aware of. And so I'm optimistic that our ragtag band is going to be able to infer a timescale of vascular plants. And I am excited about the prospect of doing so. Eyes wide open to the challenges of the method. And thank you very much for your attention. So is your question, how do we empirically know what rates we should put in this analysis for fossilization, speciation, and extinction?
Because intuitively, I would feel it's kind of unlikely that we happen to have a series of fossilization events on the same lineage. And we usually are not so optimistic about to say this fossil is the ancestor of something rather than close relative to its ancestor.
Yeah, I'm going to try to maybe answer your question in parts and let me know if I don't quite get it. The first part is there is often a bit of a knee-jerk reaction that this is weird. What are the chances that we're actually going to sample something that happens to be directly ancestral to something that we see in the present day or elsewhere in our sample? You could have fossils that are ancestral to other fossils as well. Like all these fossils are ancestral to this fossil. They left no descendants, right? So that seems, I think, intuitively unlikely to many people. But if you crank the math, it's not quite as unlikely as you think. So if you have a process that has a rate of speciation and a rate of extinction and a rate of fossilization, there is a chance that you will have a fossilization event that is on a lineage that survived to the present. And that chance is not as small as you think, depending on the range of parameter values. If extinction is very high, it becomes very rare. But if extinction isn't very high relative to speciation, no, if extinction period isn't really, really high, and fossilization rate is relatively high, that becomes quite expected. I'll also say that this method involves a model by which those parameter values can be estimated. So we use the simplest version, which involves the same rate of fossilization, speciation, and extinction across the phylogeny. That's almost certainly a problematic simplification, and we will relax that assumption moving forward. But then those rates are estimated from the data. So what rate of fossilization, what rate of speciation, and what rate of extinction best explains your observed data, including the fossils that you have? And I didn't highlight this perhaps, but in our particular data, from the fossilized birth death, we inferred 2 fossils. I didn't mention this, but here's one. Two samples that are ancestral to other samples. So this Scolecopter, Incisophilia, was inferred as being directly ancestral to this other Scolecopterus, and this Meradiopsis was inferred as being directly ancestral to this clade. So given the parameter values, this is the distribution of sampled ancestors that was most probable for this data set. Not a lot of them, but some.
You very skillfully gave an entire presentation on model selection and biases and stuff, and you purposefully avoided the weeds of priors. So I actually had a problem similar to that, the very pull of the present kind of thing across certain groups, and I wasn't able to resolve what in the data set that I was using. So this is a really great talk for me. Yeah, that makes too.
Yeah.
I was wondering how you went about working through specifically prior space for different types, for example, for the nodes, right, or for the tree prior. How you ended up deciding those, or if you just left them as defaults or uniform, for example.
Okay, so I will maybe decompose that question into 2 classes of priors. There are a bunch of priors that we didn't care much about, and to be a little bit less flippant, there's a bunch of priors that we're quite confident we're going to be able to estimate well. Parameters are going to be estimate well. We have a lot of prior information. We're not concerned, and that includes, for example, the substitution model. So we didn't even really think too much about the parameterization of our substitution model. And I'm being a little facetious about that because that's actually not entirely true. One of the challenges about these models, which I didn't talk about, independent of the structure of the model in terms of the actual characterization of our prior knowledge, is to get them to run well, to get them to mix well. And so there are some tricks of parameterization that don't change the underlying biology but allow the model to sample better. But I think what you're really interested in are cases where the priors really matter for the conclusion. And that's the thing that's actually the core of what we're struggling with. That's the things that are in these two boxes. So these are different sets of parameterizations that come with priors that do have an effect, or we're concerned have an effect on our data, on our inferences. And one of them very, very much does. So these are two different priors. There's a uniform tree prior and a fossilized birth-death tree prior. And this one has a few other components. So it does have a prior on the speciation rate, a prior on the extinction rate, and a prior on the fossilization rate, which we did work with to try to achieve things that seem to be coherent. But we're not at the stage yet where we are really focusing on that sub-level. We're still in the stage of the broader model structure, the broader inferences implied by the model. But prior sensitivity is hopefully one of the big messages that I'm trying to get across here. In certain types of problems, and divergence time dating is chief among them for evolutionary biology, the problem itself is extremely prior sensitive. There's no way around it. We're used to priors that you can get around by adding more data. There's no amount of sequence data, and really very little amount of molecular data that we can add to this data set that will reduce the impact of the priors, because those data don't tell us how rate and time are decomposed. That decomposition, which is critical for the divergence times, is coming entirely from the model, from the priors. This is a different flavor of priors than we're used to working with in Bayesian phylogenetics, where you can overwhelm an uninformative prior with more data. You can add as much data as you want. It's not going to change the impact of those priors. So it's all about then the biology of the priors. It's all about them genuinely reflecting your prior understanding rather than attempting to have some uninformative prior. That's not possible. Is that sort of okay? Yeah, Dan.
This is just more of a general sort of philosophical question about what it means to assess model adequacy the way you're doing with posterior predictive simulations in that sense. And so I think it's really interesting that you see, convergent or congruent patterns between different models of essentially relaxing or introducing or relaxing correlations across sites in rates, for example. So you see between the fossilized birth-death process and the non-fossilized birth-death process. But I guess the question is, so that's interesting. There's obviously something going on that needs to be explained there. But the question is, though, where do you draw the line in terms of what actually constitutes adequacy here? So clearly you're getting very divergent results here, but even independent of that, the question is, what does it mean to fail to overlap zero in that sense? So you'd never, with any complex data set, my expectation would be it's not going to match, but how good is good enough? So, taking that right there, that could be, if you hadn't used the fossilized birth-death process and you're just looking at that information right there, it doesn't provide a way of gauging the value of that difference from zero in terms of how useful the predictions are of the model, right? So I'm just wondering, what's your perspective on how we should go about thinking about the utility of posterior predictive simulations in that sense, in terms of how close to zero do we actually need it to be to say this is bad or this is good?
Yeah. Okay. So there's the classic line probably everyone has heard, all models are wrong, but some of them are useful. This doesn't tell us that this tells us the model is wrong. It doesn't tell us whether it's useful or not. And like Dan said, if we slid these a little bit further this way, would we think suddenly it's okay? Or a little bit further, is it okay? We don't know. And I don't think there's really there's no objective answer to that question. I think in the literature, people tend to be pretty loose about it. They'll say a model is adequate if it is able to produce the data of the type observed. So if these curves have a non-zero overlap with the data, you might say the model is in some way adequate. That's a really low bar, and it still doesn't answer your question. Is it useful ultimately? But I will point out that's not really what we're doing this for. We're using this as a tool of diagnostics. We're not trying to say that this model is right, but we're trying to say that this model is better than this other model for reasons that we can understand based on the information that we're getting here. We can also say, for example, that these models are identically bad even though they have fundamentally different inferences. And so even the whole question of model adequacy isn't really at play here because the data that the model produces is basically the same. So to maybe answer your question, I don't think there is an answer to the thing that you're looking for, but that's also not really what we're using this for. So the question is, it relates to so how much gain can we hope to have from increasing our amount of morphological data? If we have perfect morphological data, we'll know exactly the relationship among the fossils and the rest of the tree. And we'll know the branch lengths in terms of number of changes, but we still will have no additional ability to pull rate and time apart. So they help, but they don't help with the underlying big problem.
No, that's true, but the confidence with which you can place the fossil on the tree should change your age estimate significantly.
I think practically speaking, that I would agree with you. So if we were really sure from our morphological data that these guys have to be on this branch, then suddenly we have to pull this node back. But that's only, I would say it's very much an edge case. Still, for any given branch, the only direct inference we really have is about the amount of change. And you still have an opportunity to have a huge amount of time in a very short branch, in a very fast branch, or a lot of time in a very short branch. So it certainly helps. I think that will be a main way in which our inferences improve, particularly in their precision, but the big underlying problem is still there. Yeah.
I had a similar question about how confident you are in so you're combining living texts that have molecular and morphological data with fossils that only have morphological data, right? So how confident are you of the fossils on the tree, or do you just go with where they end up? The only thing that I've ever done like this, the fossil had a combination of characters that don't exist in modern or living taxa. So one place that you end up is almost as good as the next best and the next best and the next best. There's 3 or 4 places, and that spans 50 million years or whatever. And so where you pin that on the tree would.
Yeah.
So do you just go with where the fossils end up in these analyses, and the confidence doesn't matter?
Yeah, so the question, if I can paraphrase, is like, how confident are we in this phylogeny, in the actual relationships, especially given the fact that fossils have relatively little data? It's only morphological data, and it's often highly incomplete, because there's not that much information available in a fossil. And the answer is that you may have noticed I have not put any posterior support values on this tree. So none of our trees have much by way of support at all. We really have very little information about the specifics of the phylogeny. But the inferences of time are integrated over all possible positions in the entire posterior sample of all of the fossils. And so it's not simply the case that we take the best one and make all of our inferences downstream from that. The inferences are accounting for the uncertainty in the phylogeny, which is substantial. Is that okay? Yeah.
Other questions? Okay, I asked the last question. So it's I understand less about a theory, but I think I'm more interested in empiric aspect. So I first heard about this chip dating method just a few years ago after we did a molecular dating and without being aware of it. And so then I looked into the literature. My first reaction at the time was that theoretically this is a beautiful method. But I was most worried about its application, its practice. So from seeing your work, what I'm questioning is that the software, the algorithm you're using, how well developed do you think it is to integrate the morphological model into a model that was largely molecular, designed largely for molecular data set?
Okay. So the question basically is, what's the adequacy of the software? So we did all this in RevBase, and RevBase is relatively new, and it is still definitely glitchy. So there's still just straight up bugs, things that don't work. But there's also the fact that a lot of the models haven't been explored that well. But I would say in general, those problems are not probably going to be important for very many applications. By the time people get their analyses working, they have resolved the actual software side of things. But the remaining problems are about what the software is being told to do. and which is what model you're running. And that, the software can do its job perfectly, but you're still going to get a weird answer if your model is weird. It's like that joke about computers, right? They do exactly what you tell them to do, even when you really want them to do something else. So that's definitely the case with these models. The software can work perfectly, but you still have an issue because your actual model is weird.
I imagine this software has been used by some other research groups analyzing different taxa. Do they get a similar experience as you did?
No. So it has been used very little. I should have a better answer immediately off the top of my head, but I would say there's been maybe 3 studies that have used this so far. Maybe that's a little bit underestimate, but there haven't been very many. There's been a handful, and they've often done something like we described, involving some sort of model assessment. But I do think that we are in early days now where we should be cautious about any of these results. But I want to emphasize that the fundamental issues that we're facing here are shared with the classic approach, the node-based approach, except they're hidden a little bit because we're just attaching those curves on the edges of nodes. So you don't quite see the same range of behavior that we see. The priors are more informative. The time priors are more informative in those approaches. But your result is still equally dependent on those priors. So if those priors are off, your result is likewise going to be off.
Okay, so first time you finish really early ahead of time. So thank you for all that time.